recap from last week. Last week, I briefly covered one of the iGEM 2019 teams. It was the high school Lambert team. And what they did is they created a biosensor that detected the parasitic roundworms, among a few other different types of parasites that are very, very common within developing countries because the water that they drink is not properly filtered. So I wanted to include a few notes from last week, some stuff that we learned and that I discussed with regards to these presentations, how they're structured given what the judges expect from our team while we're giving these presentations. The first is Lambert in their dry lab, which was something that I'm trying to do this year, built their own fluorometer device, which brings into question, should we build our own or should we borrow someone else's schematics, all the other research already done, and then build upon that? I've said this before, but I'm of the opinion that we should do the latter. We should build off pre-existing designs because that is much easier, especially since this is not like the focus of our project. The dry lab isn't, it's more the wet lab stuff. The dry lab is just kind of a flair that you can add to your project to make the judges like it better. So I also wanted to note that the general sequence of these presentations are broken down into this sort of structure. So first you want to name the problem or some sort of exigency that is happening in the real world. You want a problem that is probably related to your hometown, home city, home country, all that sort of stuff. So you have people in the field close to you that you can ask, or just laymen that experience this problem on a daily basis that you can do research on and ask them, would they be willing to buy or use your device to fix that problem? Judges really like the problems that directly relate to you because you have a better ethos, right? You've dealt with this problem in your own environment. So you have some sort of experience with it and you have an exigency to fix that problem. Then they moved on to solutions. So you want to provide usually a synthetic biology solution to whatever problem that you're trying to solve. There is many, many different ways of doing this, but theirs in particular was creating a device that allowed them to sense roundworm parasitic contamination within water samples. And so what they did is they created filters here in their dry lab section that allowed them to separate and lyse the parasitic roundworm cells. So they could test the DNA to figure out if you had that contamination. And in order to do that contamination, the testing for the contamination, they use something called an RNA toehold switch. This is a diagram of a toehold switch. It looks a little bit complicated, but rest easy, it's not actually that bad. So what it is, it's just before it's structured like this, it's a linearized piece of RNA. And so when this RNA begins binding with itself, there's going to be a little extra tail end called the toehold. And this is where your activator RNA, whatever you're trying to test for, is going to bind at. And this is just a continuation of that sequence. But this continuation of the sequence binds to the complementary, to itself, right? Because it's a single-stranded RNA. So it has a complementary strand within this RNA. You can notice here that the AUG, the start codon, and the ribosome binding site are not part of that overlap. And that is because once this activator molecule right here, the reverse part of that RNA, binds to this, it's going to want to continue binding to its complementary base pairs up this strand. And as it does that, this segment right here is going to become unlatched. And so as this comes apart, this ribosome binding site finally gets exposed along with the start codon. And this right here allows the ribosome, when you're doing translation, because this is RNA, to finally attach to that ribosome binding site and translate and make proteins for whatever gene that you want to translate or express. And so this whole system can be considered a repression system. It's a regulatory system that allows you to repress whatever gene is downstream from the toehold itself. Next in the sequence of slides were improvements. And so this is where you're going to list out what parts that you characterized. So if you used parts in the registry, used schematics from other teams, you're going to show how you improved those schematics or parts. Also, here I recommend detailing your new parts if you created any new parts, whether that is through point mutations, site-directed mutagenesis, all that sort of stuff. You detail that there. Next, they detailed dry lab. Dry lab was a very big part of their project due to they created two things. They created that filter, and they also created the homogenizer that allowed them to lyse those cells. That's pretty much just a centrifuge or a vortexer that spins the cells rapidly and then breaks them down so you can extract the DNA. So, of course, you wanted to start with the more of the synthetic biology stuff and then get to the dry lab because the synthetic biology stuff is more important for what we're doing in iGEM. Finally, they finished their project, their presentation with modeling. Modeling is very, very important. It's one of the categories for silver that we have. I believe you can do really two things here. You can model the kinematics of the whatever device that you're using, how well it works, what the fluorescence is, how good the intensity or response was. I think that was what they did in their project. They measured how quick the response was, how intense the response was when their activator DNA or RNA binded to the toehold switch. So modeling is there, and then finally, I want to emphasize they finished their presentation with integrated human practices. So human practices that directly related to their project, something that they went out to go to do that builds upon what they're doing for their project, not just something not related, right? They wanted something that directly related to what they were doing. I believe they visited, I think it was the Congo, they visited Africa, and they asked the people how common this parasitic roundworm infection was so they could gauge the exigency of the problem. So yeah, I did all this. The part that they characterized was the promoter. I believe that the promoter that they used within the gene was too active, so that led to leakiness, so they got false positives. So what they do is they used a moderate promoter in order to make that less leaky. And that is all I have. This is the part that they used. As you can see, it's not very complicated. So you have your toehold, and you have your promoter, and then you have your GFP, reporter protein, and then your stop codon. So this part of the system is not that complicated, and it's something that you can typically expect from a high school team, which is what this team was. So next I want to move on to Golden Gate assembly. This should go by, I would say, pretty quickly. I'm going to briefly go over this. In the past, I've covered this little thing right here in the Gibson assembly protocol. But previously, we covered 3 different types of assembly. The first was standard. In standard assembly, basically, you're just putting one insert into one backbone. And that has a few disadvantages because you can only do that one single insert. It requires gel purification. So after you properly ligate the 2 parts together, you have to purify it in order to get your ligated part away from the non-ligated parts. And it also, because of this gel purification, among other things, it has a very low success rate, especially compared to a few other assembly protocols. 3A assembly was an improved standard assembly method. It uses 2 inserts. However, you need 3 different antibiotic genes in order to properly screen for your ligated parts after you have them finally ligated. And this is because in your mixture that has all these different parts, some are going to be ligated properly with all the different inserts inside the backbone. Some are just going to have, they're just going to be linear, and you only want to pull out the properly transformed bacteria. So the next thing I covered, though I don't really mention it here, was Gibson assembly. That I believe was 2 lab meetings ago. Gibson assembly allows you to ligate together around 6 fragments. And the main disadvantage to Gibson assembly was you had to design the primers. And these primers were anywhere between 25 and 40 base pairs. And that therefore leads to some expensive primers that you have to synthesize with external companies. So that's the main disadvantage with Gibson assembly. And so now we can kind of move back to something that I view a little bit less complicated than Gibson assembly, and that is Golden Gate assembly. This was discovered in 1996. And some of the advantages of this assembly protocol include the ability, just like Gibson, to ligate 6+ fragments together. Though unlike Gibson, you can go up like pretty far depending on how many reaction steps you do. I think I saw online some people ligated up to 24 plasmids at a time, or 24 fragments at a time, which is definitely crazy. I wouldn't recommend doing that though, because when you're ligating that many fragments together, the efficiency is going to be greatly decreased. on an exponential order of magnitude. So stick around 6. Typically, when we're working with iGEM, we use around 3 or 4 max, I would say. I did not mention this in the presentation. I'll get this a little bit later in more detail, hopefully, if I don't forget. But I would say the main benefit of this procedure is its seamless and scarless nature. So with 3 assembly, when you're ligating these parts with different restriction sites on the end that have sticky ends, when you ligate them together, they're going to form scars. So when the restriction sites combine together, that restriction site you can't really use anymore because it's a combination site. So with Golden Gate assembly, though, there are actually no restriction sites present within your finalized plasmid. I'll get into this a little bit later. I think it's like the next slide. But you only use the restriction sites. It's just one type of restriction enzyme for a single step. And this makes it a very, very powerful tool because your products are just products in their entirety. They're pure. They don't have any of these external sites that might mess with whatever you're trying to transform with. Also, an improvement compared to Gibson assembly is you don't need 20 to 40 base pair overhangs when you're doing the PCR preparation step. You only need 4 base pairs, which in turn makes the whole process a lot cheaper and a lot easier to design. And I'll get into that a little bit more in the next slide as well. Hand in hand with the seamless and scarless nature of this protocol is the efficiency. So when you start at this procedure, you're going to have your backbone with your restriction site in it. They are the exact same restriction sites using one single restriction enzyme. And you have your fragments with these 4 base pair overhangs on the end. Of course, those base pair overhangs will correspond to the sequence at which you're trying to ligate the fragments together within your fully functional part. So why is it close to 100% efficient? This is because when you're cutting the parts, and like I said, when you cut them within the part itself, there is no restriction site. When the part is fully ligated together, the restriction enzymes already present in the solution can't cut it again. It can't go in reverse. So what happens is you have your unligated parts in the solution, and as they're being ligated together, it's a one-way street. So it doesn't go backwards. There's a 50-50 chance that the ligation is going to work or just recombine to what it was originally. I will get into this in the next slide just to make the visuals more, to make them more understandable, right? But I want to get into the disadvantages now. So the main disadvantage is that if you have a type 2S site in your fragment, that will completely mess up the procedure. So basically what happens is you have your parts with the primer overhangs. And if you have that restriction site in the center of the part, then the endonuclease is just going to come in, it's going to cut your part in half, and then you're not going to be able to use it. Or you can use it, but it's more complicated. So for our intents and purposes, you just can't use it. And also disadvantages, like Gibson assembly, you need unique flanking sequences, so unique primers at the ends of these fragments. If they aren't unique, they're going to combine in random orders, and that in turn will make a non-functional part. So here is where most of the clarification and explanation is going to occur. So like Gibson assembly or any of the other assembly methods, Golden Gate assembly, the main purpose is to ligate fragments together in a sequence. So you want your promoter in front of your coding sequence. You want your ribosome binding site in front of your coding sequence. And so to do that, you have to use these assembly protocols. So here I detailed the nature of the seamless and scarless. So here is your recognition site, your restriction enzyme site for BSA1, which is one of the more common restriction enzymes that you can use for this. And then here is your part of interest. I guess your part of interest would be after this. So these are your primer overhangs. If you can, I guess, use these diagrams hand in hand with each other. So restriction site, restriction site, restriction site. And when this restriction enzyme comes to recognize these sites, unlike EcoR1 and other type 2 restriction enzymes, it doesn't cut down the middle here. It doesn't cut like that. And that's because it has the S prefix, and that denotes the fact that it goes one down and then begins cutting. And so that's very, very useful because it cuts downstream, the restriction sites don't even have to be in your part. They can just be outside of your part. You cut it, it leaves that overhang that you can use. You ligate the parts together like here. And then your restriction enzyme site's just gone, right? You don't have to use it. In sequence, how this procedure works is, first you want to get your vector, so whatever you want to, your backbone that you want to insert these parts into, and you want to make sure that it has these recognition sites for BSA1 or whatever enzyme that you're using. And of note here, you're going to have a forward restriction enzyme site, and you're going to have a reverse restriction enzyme site. And that is because we want only this backbone, right? We want this backbone without the sites themselves. And so we do this when this gets cut, it goes downstream. So here, and it cuts like this. And you can see that right there. And same thing right here. So what happens is this whole segment with the reverse and backwards restriction enzyme sites just leaves the mixture, and it's no longer used for the reaction. So then you just have a linearized plasmid backbone with your design, because you designed these primers with these designed sites that you created that you can use to combine with whatever parts that you want. The next step in this sequence is creating those parts that have the primers, the sites that you're working with of interest. These things are called flanking sequences, and these are added via PCR. I covered this in the Gibson assembly protocol, but if you're not familiar with how to design these primers or with how to do PCR, I included 2 links at the bottom, one for what PCR is and then one for designing the primers themselves. Note though that this video is for Gibson assembly, though it is pretty similar. Just keep in mind that when you're designing them, you're not designing them 30 to 40 base pairs long, it's just 4. So yeah, you design your parts of interest, hello, and these fragments are going to have these primers attached to the ends, and in turn, depending on what your sequence that you want, they're going to match up. So right here, this flanking sequence is going to correspond with the flanking sequence of the backbone, and likewise with these intermediate like junction parts. And then finally, this part right here combines and matches with the flanking sequence of the backbone because you designed it that way, right, through PCR. After you've properly got all these parts flanked with your primers, next, BSA1 endonuclease, you don't have to use this restriction enzyme. It's one of the more common ones, though, comes in. The endonuclease comes in, begins eating, like I said, the downstream from the restriction enzyme sites, and that leaves a sticky end overhang that can combine with these parts. And then after the hydrogen bonds begin forming between these complementary nucleotides, then T4 DNA ligase comes in and begins combining and ligating that phosphodiester backbone so you can get a fully solidified part, which is seen here. Okay, so I'm going to move on to one of the main, I would say, disadvantages to this procedure, and that is if you have a restriction enzyme site within your part or fragment of interest. And the reason why this is a problem is because I mentioned this previously. If you have the BSA1 restriction enzyme site present within your part, when you're cutting it with the BSA1 endonuclease, it's actually going to cut your fragment in half, and in turn that can't be used within your fully ligated plasmid part. To check whether or not you have these restriction enzyme sites within your fragments, I recommend using this link here. Here you can, I think it brings you to NEB's page. It's just like an online calculator that they have. what you do is you insert your sequence and what enzyme that you're using, and then it will tell you whether or not you need to do some sort of extra procedure to get rid, say, site-directed mutagenesis, to get rid of your BSA1 site within your fragment. So you really have, at least that I came up with, 3 options to fix this problem, which is called domesticating your fragments so they can be used for Golden Gate assembly. The first option is just to choose different parts, and that is definitely one of the easiest of these three options, but it solves it quite easily, right? Because if it doesn't have the BSA1 site in it, it's going to work perfectly. Option 2 is to use site-directed mutagenesis. If you're not familiar, I'm not going to go into that here, but I included a link to a video covering site-directed mutagenesis. And basically what you're doing is you're silently mutating that site so it doesn't correspond to complementary recognition for when the enzyme comes in. And by silently mutate, I mean you're just changing the nucleotide to where it has the same amino acid, so it still codes for the exact same thing, but it doesn't change the functionality of it, but in turn, the enzyme can't recognize it and therefore doesn't cut it in half. So the third option, which I don't fully understand myself, but it is to use this internal site as the junction between 2 fragments. So this is your fragment 1 and this is your fragment 2. And what you're doing is you're using this as how you combine the 2 parts together, so the overhangs. So the site within the fragment is actually used as that overhang for that junction. And then while you're doing this, you can actually use site-directed mutagenesis within your primers, kind of, to where one won't recognize it and cut it again. Yeah. This is a pretty simple slide, I would say. Here, I just detailed the protocol of which you do this. It's not a very complicated protocol, especially compared to like something like mini prepping or something like that. Pretty much, you just have a few ingredients, a few solutions, buffers. Those are your primered parts and your insert vectors. Of course, you want to primer them and make sure that your insert vector has those restriction enzyme sites already present in it. I believe you can either use parts that are linearized for this process, but iGEM itself actually recommends having the parts already present in plasmids. So if you're going to have a plasmid with the BSA1 sites surrounding, flanking your gene or fragment of interest within this fully cylindrical plasmid. And then you're also going to need the Golden Gate assembly buffer just for the enzymes and T4 DNA ligase to work properly, and your master mix, which includes your enzyme and your T4 DNA ligase. And finally, some nucleus-free H2O, or I guess for us, it's called Millicue water because we use the Millicue machine. You calculate the volume of fragments in water here. That's just a link to where I pulled this from. As you can see, it's, of course, like all these procedures, dependent on your concentration of DNA. So you only want so many nanograms of each fragment within your solution. Typically, you're going to want these as equal molar fragment ratios. So you can, so one part doesn't combine and it doesn't make the process messy. Of course, these are stable. And then this nucleus-free H2O, the volume of that just changes depending on what your concentration of DNA is. After that, you incubate samples thermocycler. I included a suggested assembly protocol here. Just for that disappears, for the 1 to 4 insert parts, you do it 37 °C for 1 hour, and that allows the restriction enzyme to cut the parts, and then finally that ligation step at the end. And then, of course, the complexity increases as you increase the number of fragments that you want to ligate together, like all these assembly protocols. Pretty much it just changes though, because you just ramp up the amount of cycles that you do in the PCR machine. So instead of just doing 1, you do 30. And that is all I have for this. I wanted to include some external resources for your viewing pleasure. I included a quick workflow video, I believe from NEB. They had a decent video where it detailed the procedure that I just explained when you're doing Golden Gate assembly. I also included this is actually a really good explanation from iGEM itself. They have a YouTube channel, and they made a Golden Gate assembly video. I think it's just a seminar person discussing, and that's where I mainly learned this material. And then I also included that assembly wizard that I mentioned from NEB that allows you to insert your sequences, make sure they'll work properly, and it allows you to design your fully functional, your part at the end.